Okay, let's continue to check our laboratory exercise 5, cell structure and function. This particular model is what we use in the A&P lab for animal cells. This is an animal cell. If we were in regular biology, we'd look at the animal cell and the plant cell, and they're quite different. But let's look at the animal cell since we are talking about humans. Let's look at the different parts. Now, I'm going to start from the outermost, and this is the membrane that lines the outside of the cell. Is called the cell membrane. It is a phospholipid bilayer. It's got phosphates and lipids, some chemicals called phosphates and lipids, bilayer, so it has a two-layer thickness there, and it will not allow any water-based fluids to go through. Nothing passes through it unless it's lipid soluble, or it would move through if it were dissolvable in a lipid, but not in water. But anyway, it's called the phospholipid bilayer, the cell membrane. Okay, the cell membrane, as we're going in the cell membrane, you have all these canals, and things are moved in and out of the membrane by a form of active transport called endocytosis, exocytosis. To move into would be E-N-D-O, cytosis. Cyte is cell, C-Y-T, osis, the process of, endo, into. Endocytosis is when this vacuole forms. This vesicle forms around the object and brings it into the cell. It goes through the cell membrane. This is a way the cell membrane can allow things to be brought in that will not dissolve in fats or lipids. Okay, let's go through this. So this is the cell membrane. Everything floats around in what is called cytoplasm. So the definition basically of cytoplasm is all the contents of the cell from the cell membrane all the way to the nuclear membrane and the nuclear envelope. So all of this is cytoplasm, has all these organelles floating around in it. Okay, so cell membrane, cytoplasm. In the cytoplasm there is a fluid called cytosol, and that's what everything's floating around in. All the contents is called cytoplasm. The fluid is called cytosol. Okay, this structure here that looks sort of like a kidney bean is a mitochondria, and those little channels there are called cristae of the mitochondria. The mitochondria is the area that you have the most ATP produced, and that is what we use. It's almost like our currency for anything that uses or needs energy to happen in our body. We need the currency of ATPs, adenine triphosphate, and they are made, most are made inside of the mitochondria. Now, we have to have oxygen for that to make enough ATP, or for the process to continue in the mitochondria. Okay. Mitochondria. Mitochondria, there is a whole one. This is a cross section of one. Alright. This is a vacuole or vesicle. That's also a vacuole. Now, this is a different kind. A vacuole usually carries water. A vesicle could carry water, but it can carry other substances that are brought into the cell. These are called vesicles also, or Golgi bodies, because this structure is part of this inner membrane system. These are all interconnected with that cell membrane. The Golgi apparatus, its function is to package and modify proteins. So it takes proteins and does a little change on them, and it repackages them and sends them wherever they're needed. Okay, the Golgi apparatus and the Golgi bodies. Alright, so we have this inner membrane system here. We talked about this is sort of an inner membrane system, the Golgi body or Golgi apparatus. And then this inner membrane system is called the endoplasmic reticulum. Endoplasmic Reticulum, but ER for short. Now, if you were taking a lab practical and you were typing out answers or writing out answers for me, I would want you to spell out endoplasmic reticulum the first time, and then the next time you could abbreviate. Okay, there's two types of endoplasmic reticulum, and one has these little white dots connected to it and some doesn't. These little white dots are ribosomes. The endoplasmic reticulum with the ribosome is called rough endoplasmic reticulum. Gee, it looks a little rough there. Rough endoplasmic reticulum because it has ribosomes attached. And then this is smooth endoplasmic reticulum. Now, the endoplasmic reticulum intermembrane system, its function is to transport materials through the cell, but the rough endoplasmic reticulum with the ribosomes attached play a role in protein synthesis, making proteins. Your genetic material there codes for proteins that you need to build new parts and to live. So that process of making new proteins is carried on with the help of the ribosome, and so this endoplasmic reticulum has ribosomes attached, so it's called rough endoplasmic reticulum. There's a little bit of lipid synthesis here too, but the majority of the lipid synthesis, or the making of certain fats that we do need some fats, and those are made by the smooth endoplasmic reticulum. So smooth endoplasmic reticulum, lipid synthesis, also helps to break down drugs and a few other little things. And then the endoplasmic reticulum with the ribosome attached, the rough endoplasmic reticulum, a role in protein synthesis and lipid synthesis also. Okay, so vacuole, carrying water, probably this little gray object. The orange, the mitochondria, we nickname those the powerhouse of the cell because ATP production, and ATP is used for energy. Now we have this little object here. It's also a type of vesicle, and it holds digestive enzymes. And those enzymes do it just like it says. It helps to break down substances so that either they can be gotten rid of or we could recycle the parts, because we're recycling machines. And that lysosome is what that's called. The yellow is a lysosome. There's a cross section of a lysosome. There's another cross section of a lysosome. There's another. The lysosome has digestive enzymes, and I think of it like a little Pac-Man where it envelops a microorganism or a worn out cell part or worn out cell so that it can break it down, recycle the parts, and get rid of the leftovers or get rid of the junk. And this larger structure is called the centrosome, and inside of the centrosome are the centrioles, these rod-like structures where you have microtubules or spindle fibers formed. They go to opposite ends of the cell, and it plays a role in mitosis. We're constantly needing replacement cells, so that cell has to divide. But before it can divide, it has to go through these steps to replicate everything inside. But anyway, these centrioles play a role in mitosis. Now, over on this side of the model, we can see some smooth endoplasmic reticulum, and then there's some rough endoplasmic reticulum, and we already talked about that. Okay, let's look at the nucleus. Now, all of this is cytoplasm. Now, in the nucleus, that fluid is called nucleoplasm. All the contents of the nucleus, nucleoplasm. So you have a nuclear envelope and a nuclear membrane. It's very porous because things have to leave that nucleus and go out to the cytoplasm. And ribosomes is one of the things that come out of the nucleus, because in there is the nucleolus spelled a little different than the nucleus. Be careful on that spelling. Okay, the nucleolus makes ribosomes, so it plays a big role in protein production. Ribosomes, we talked about the ribosomes. They're free-floating in the cytoplasm. They're attached to endoplasmic reticulum, and some can be found in that nucleus because that's where they're made. The role of a ribosome is protein synthesis, okay, the nucleolus. Now, the pores here, again, allow for those ribosomes to come out in different structures. Okay, these little coiled-looking structures there are representing chromatin. Chromatin, or chromatin, as some pronounce it, is the decondensed or stretched out chromosomes. Now, these look a little condensed to me, so this cell may be getting ready to divide because chromatin or chromatin will condense to where we can see the chromosome. Once it's condensed and we can see the chromosome, then we call those chromosomes. So you can call them chromosomes because we can see them. With the compound microscope, if we can see them, it's getting ready to undergo mitosis. So chromosomes are chromatin, decondenses chromatin, condensed would be chromosomes. And the chromosomes, yes, carry the genetic material. To every cell in your body that has a nucleus, that has chromosomes, has genetic information to make a whole new you. If we can manipulate that genetic material from a stem cells to a dedicated cell and grow all the parts that would make a whole new you, then we could, because all the information is found on those chromosomes. Those chromosomes give the instructions for making proteins. And proteins is the basic unit that all macromolecules, all the things that you need to grow a whole new you is found right there in that genetic information. It's the code for different types of protein. Okay, have we missed anything? Don't think so. That is the cell.